development. This is the topic, is the need of the hour, and apt in the present global scenario for scientific advancement. The growing needs on health issues triggering the scientists in pharma industry to combat day-to-day emerging challenges in the world. Now, I take an opportunity and pleasure to introduce today's first session speaker, Dr. Devapriya Kumar, going to give lecture on topic A and ML in drug design. Dr. Devapriya Kumar is presently working as Associate Professor in IIIT Hyderabad and also currently holding the post like IIT Hyderabad Academic Head, Technology Innovation Hub at IIT Hyderabad, Head Center for Computational Natural Sciences and Bioinformatics, Coordinator for Sponsored Research Cell Faculty in Charge, High Performance Computing Facility, and also chairing different lectures, distinguished lectures committees. Devapriya Kumar has completed his MSc MPhil in Pondicherry University, PhD in Computational Chemistry in Pondicherry University, and also in collaboration with IICT Hyderabad in Computational Chemistry. Priya Kumar has received a number of awards and recognitions like Chemical Research Society of India Bronze Medal, Visiting Fellowship from INSA and the Royal Society of Edinburgh, Distinguished Lecture Award from Chemical Society of Japan, Indian National Science Academy INSA Young Scientist Medal in Chemical Sciences, Young Associate of Indian Academy of Sciences, Innovative Young Biotechnologist Award from DBT, AICTE Career Award for Young Teachers, and Young Scientist Research Award by Department of Atomic Energy, and also awarded to participate in meeting of Nobel laureates and young research and science students held in Germany. He has received a gold medal for academic excellence in MPhil and MSc in Pondicherry University. Sir has received number of grants for different projects like CSIR, DST SERB 2 times, INSA, DBT 2 times for 2 projects, AICT, etc. He has organized a number of scientific meetings in machine learning, computational chemistry, in biology and medicinal chemistry at national and international conferences, and also conducted different winter schools. Now, I take it privilege to invite once again Priya Kumar Garu to start his lecture. Thank you very much, sir, and thank you very much the organizing committee for giving me this opportunity. Sir, please.
Yeah. So thank you very much, Professor Devapriya Garu. So, thanks for a very nice introduction. Also, I should thank Professor Sridevi and then Professor Pooja and everyone in the organizing committee for inviting me. So online is getting a bit too much, but, still it's nice to meet people, even when we are sitting at home. Okay, so I will share the screen. You can see my screen, yes?
Yes, yes.
Okay. Okay, so today I'm going to talk a little bit about introduction to machine learning methods or artificial intelligence machine learning methods for drug design. Okay, so what I will do is I'll spend maybe around half hour in introducing the concept of machine learning, in as simple terms as possible, right? And then I'll take the rest of the time in showing some case studies in terms of how we can use artificial intelligence and machine learning methods for designing drug molecules. Okay, so as Professor Purnavati said, so I'm from IIIT Hyderabad, and I'm from a center called Center for Computational Natural Sciences and Bioinformatics. Okay, so you can go to these websites to get more details on the kind of work that we do in the area of machine learning and also in other traditional science areas. Okay, so this is how I think we will take the talk. So I will give a very general introduction of what is the relevance of artificial intelligence method, what is the relevance of machine learning in general, and then in the context of molecules, okay, in the context of the drugs possibly, okay, and then we will try to understand again in very simple terms what we mean by machine learning, because everybody talks about artificial intelligence, everybody talks about machine learning, right? So we will understand in simple terms, at least one layer of first layer of qualitative understanding of what we mean by machine learning, right? So we will spend some time on those, and then I will take some 3 examples. Okay, so one is a protein structure prediction using artificial intelligence methods. Okay, so this is not a work that we did, but but this work is very, very popular. It made a lot of news because they did extremely well in terms of predicting the structure of proteins. And then the next example I will take is like simple molecular property prediction. If I have a molecule, I want to predict the properties of those molecules. Okay, so I will take that example. And then finally, if this, what we call inverse molecular design. So this is, a little bit, more interesting than others, especially for me, because, we do things, quite differently from what we do in traditional ways of designing molecules. Okay, so that's the case studies, and depending on how much time we have, depending on how fast we go, we will cover as many as possible. And then please feel free to, I do not know how it works, but if you want to ask questions in between, please,
The questions may be posted in the chat box, sir.
Okay, so someone will
coordinate you, sir.
Okay, that's
Pooja
good.
will coordinate.
Okay, thank you. Okay, so this slide or this particular picture is something that you may have seen in the last 2 days in some form or other. Okay, this is like a regular drug discovery process, right? I'm sure one of the speakers must have showed a slide, something like that, right? If not, it's okay. We will we will see a much simpler picture which is necessary for this particular talk. Okay, so the idea is very simple. Once you have a disease, okay, what you want at the end is a drug that can be used to treat the patients with that particular disease. That's basically the cycle, right? So it involves understanding of the disease, and it's a long process. It takes a lot of time, right? It takes a lot of time, and then it takes a lot of money and all those things, right? So I'm sure somebody must have mentioned. So let's look at exactly the same thing, but in a slightly simpler version. Okay, so what I'm showing in the next slide is exactly the same thing, but much more coarse grained, much more with much less details. Okay, so let's say I have a disease. Okay, so for example, now we are all sitting at home and doing all this because of the COVID-19, right? So let's assume I have a disease, okay, and then what I want, or what we want, is a drug that can treat this particular disease. Okay, so I'm not talking about vaccine, right? I do not know anything about vaccines. We don't work in that area, at least at this point of time. Okay, so once you have a disease, the second thing that we want to do is to be able to identify a target. What do you mean by identifying a target? So identifying a macromolecule in the system, right? Usually a protein. Okay, so when you target these protein molecules, you intervene in that biological pathway which is responsible for the disease. Okay, so for example, most of you might have read that there is this spike protein present in this SARS coronavirus 2 interacts with the angiotensin converting enzyme 2 in the cells present in lungs, right? And then that's how the virus enters the host cells. Okay, so for example, your ACE2 could be a target, where if you can have a drug which binds to that target, the spike protein may not easily interact with the easily interact with the ACE2 or enzyme, and hence the entry process of the virus into host cells becomes difficult, and hence the immune system can easily fight off this virus, right? Because the extent of infection is much lesser, right? That's basically what we mean by target identification. Identification of the target. I don't know, there's a lot of noise coming, so if switch it.
Now, okay, sir. Now I was muted all. Sir, unmute yourself. Sir, unmute.
Kumar Garu. It seems has.
It's okay?
Yeah.
Okay. So I think when you unmuted, it unmuted me also, I think. Okay, so that's basically what we mean by target identification. You identify a target, and then next step is like this is the drug design part, where you discover molecules that can bind to this target and hence treat this particular disease. So you discover small molecules. How do we do it? We use computations, we do synthesis, we do biochemical assays, we do cellular assays, we do experiments on mouse models possibly, and things like that. Okay, so we do experiments of different kind. That's what we do. And then once we are convinced that we have a molecule that actually can bind to this target and hence can treat this disease, we go for what are called clinical trials, right? So it's several phases of clinical trials, and then we test it on human beings, right? Volunteers, and then based on the results we get from here, the regulatory agencies will decide whether that drug can be the drug is safe and it actually works, and then it is marketed. Okay, so this is the usual drug discovery process, right? So I think most of you would know. I just wanted to kind of start from the same plane. So quick discussion. Okay, so now let's say we have this process now, right? So how do we normally do? There are different kinds of experiments, different kinds of computational methods, different kinds of at different levels, right? Starting from the molecular level to the organism level, we do all kinds of experiments in the chemistry lab, biology lab, biotechnology, pharmacy, pharmaceutics, formulation. A large number of these experts are involved in this particular pathway. So let's think of a scenario, okay? So where, I have a disease and then some basic biological networks that are associated with that disease is analyzed, and then we have identified, okay? So what if there is an algorithm, okay, that can take this information from these studies from biology and then identify a target, right? Without we having to do much. It just takes the information that we get in terms of our understanding of the disease itself and understanding of the pathways themselves, and then takes identifies a target. Okay, so once a target is identified, what is a target? Usually a protein molecule, right? We have identified a target which can be targeted now. So once I have a target, that would mean that getting the sequence of these proteins are not so difficult these days, right? So once I have the protein sequence, what if there is an algorithm which can take this sequence? Okay, so you can take the sequence, just the sequence of the protein, and then gives me a structure, right? Gives me a protein structure, which is very, very reliable. Like now, we use X-ray crystallography or NMR or other experimental techniques to get the protein structures now, right? So computationally we do homology modeling and then some ab initio kind of methods we do, but still we are not there yet in terms of reliably predicting the three-dimensional structures. We are reasonably successful, right? But then not always right. Okay, so what if there is an algorithm which takes this sequence and then predicts the protein structure? So once you have a protein structure, what if there's algorithm that exists that can take this protein from here, right? So basically we have from this information we got the target. Once you have the target, I get the structure, and once I have the structure, you do a de novo drug design. What is de novo drug design? Basically starting from the protein structure, right? You design molecules, or you synthesize, or you design and synthesize molecules that fit into that binding pocket, right? That's basically what is de novo design. What if that algorithm exists that can do it for you? Okay, so once that is there, right, once you propose a new molecule, right, somebody has to make it, right? So how do I make it? I need to go back to the organic chemist and then say, okay, how do I make it, right? What if there is an algorithm which says that you give me a molecule, I'll tell you exactly how to make that molecule in the lab? What if that kind of algorithm exists? And then once that is there, what if there is a robotic platform that can take this synthetic route proposed by the algorithm, and this robot takes it and then makes that synthesis, right? It gets whatever, there's some technician who will provide whatever chemicals that are needed for the synthesis to the robot, and then robot will do everything. And then, so once I synthesize, what do? I take the molecule and then have to confirm, characterize, right? So what if there is a molecule which can take all the spectra of these the product here, and then does the characterization for you, right? Without one chemist having to look at the spectra and then interpret. There is an algorithm which already does it. Okay, and then so on, and then up to the point where the algorithms exist to help you determine whether a given molecule will be successful or not in clinical trials. Okay, so what is that I have shown you now? So basically, initially we saw this particular pipeline, right? We just saw this one and then said, if one has to design or one has to come up with a marketable drug, you have to start from here and then go all the way there systematically, and that involves a large number of experts, right? So now you say that there exists a lot of algorithms which does different components in this particular process, right? So these are some of the actual examples that came out in the last less than 2 to 3 years. Okay, all these papers, you can see, all these papers that I have put there, all the titles, all of them have been published in the last 2 to 3 years, less than 3 years, all of them, right? So when I say, what if an algorithm takes a sequence and predicts a structure, that's exactly what that algorithm does. That's exactly what that paper is about, right? So that is the context in which we are talking about, drug design, where you have a drug discovery process, and then AI, all these algorithms are based in machine learning, deep learning, artificial intelligence based methods. Okay, so that's basically, all these methods can contribute, all these AI ML methods can contribute towards this particular process, and hence can make it faster. And then in the recent COVID-19 situation, we have seen a lot of examples where people have proposed, molecules of interest in terms of our hypothesis, right? For as drug candidates. Okay, so that's we have been seeing so many papers that are coming out. Okay, so that's in that context, right? You have these deep learning, machine learning, artificial intelligence based methods, right? So it's like, what are these things? Artificial intelligence is a whole area, right? So the whole thing is artificial intelligence, okay? A subset of artificial intelligence is, deep learning and machine learning methods. A subset of, I don't think I'm drawing it right, but yeah. So what I wanted to mean is, when you say artificial intelligence, it includes both machine learning and deep learning, okay? And deep learning is a subset of the machine learning methods, okay? All right, so today we know that we use artificial intelligence methods in a lot of places, right? It has come in day-to-day life, right? So these are some of the top applications that we already have, right? So like the travel, like for predicting delays in flights, and then to come up with costs of tickets, ticket pricing, dynamic pricing of tickets. And then in marketing, social media, like the other different social media platforms you have, like suggesting friends and suggesting news or suggesting articles or suggesting videos. AI is used in great way. And then in automation, we have self parking cars for a long time now, and then we are starting to see self driving or autonomous vehicles already, right? Even in India, there's a lot of research going on. In IIIT, we have a couple groups which work on autonomous driving, right? And then in a lot of other places we see, right? So today we are going to discuss a little bit of healthcare. I will introduce healthcare as a whole in terms of how AI methods can play a role in healthcare as a whole, and then we will worry about drug design a little later. Okay, so that's where the space that we're looking at. Okay, so the idea, or this picture nicely kind of gives an idea of how artificial intelligence can be used for different healthcare applications. Okay, so these in the left, what you have are the different kinds of data. These are not the only ones, right? I mean, of course, you have large number of other data which are relevant in healthcare, but just some examples of some of the data that is relevance in healthcare, right? For example, the DNA sequences, the genome, right? Transcriptome, right? And for example, the protein structures, and then small molecules, right? You have, these are some of the data that is relevant for healthcare, right? And then what is that I want? What we want is, for example, we want to do disease stratification, or cancer diagnostics, or diagnosis in general, right? Or drug design. That's what we are going to discuss today. And then protein structure prediction, right? So what we want is this is my input, or this is what I can get, or we can get, right? And this is what we want, right? The missing piece between the two could be your AI algorithms, right? So usually this is, normally this is used to represent artificial neural network, right? But in general, the AI algorithms can be your method of choice that can connect from what you have to what we want. Okay. All right. So just to give one particular statistic of how AI methods have been playing along, is seen as a very disruptive technology in drug discovery space, right? In November 2017, that's about 3 years ago, right? So in November 2017, there were 37 startup companies, mostly in the US, right? So that were there in the space of AI for drug discovery. They, have some, they were doing some drug discovery aspect and then using AI-based methods. There were 37 companies, okay? And then early this year in April, 2.5 years later, you see that number grew up to 230, right? That's a huge surge in terms of the number of companies who have entered into the space. And then this is actually from a blog. And then the author says that, I can no longer keep up with the growing number of startups using AI for drug discovery. And he stopped updating. I haven't checked recently. Okay. So that's the kind of disruption that we see in terms of how AI methods are playing a major role in drug discovery aspects. Okay, so that's the general context that I wanted to give in terms of, placing AI or understanding AI in terms of a method that we can, in terms of an algorithm or a group of methods that we can use for making drug design more, efficient, right? Okay, efficient, in terms of the cost, in terms of the time, and so on. Okay, so now let's, if there are any questions, we will, if there are anyway, so if there are any questions, please, the moderator, please stop me anytime, no problem.
Yeah, definitely, sir.
Yeah,
Sure.
thanks. Yeah. Okay, so, right, so, okay, so let's, now this is Zoom, right? So we'll also find out how many of you are still active. So let's do a demonstration, okay? So let us say that I'm going to show you an image, okay? I'm going to show you a photo or a picture, right? So it has some Chinese character written in that one, okay? So this Chinese character written on that image, right? So there are only three possibilities. This is 1, 2, and 3. The image, when I show it, it can be either 1, 2, or 3, right? I'm not showing a large number of things, different things. I'm just saying 3 different things. Number 1, 2, and 3. I took a Chinese character because, that is possibly the language possibly, least of us would know, right? So let's, that's a demo, right? So we are going to solve this problem now, right? So what I'm going to say is, okay, someone is going to show me a photo which says 1, 2, or 3 in a Chinese, and then we have to identify whether it's a 1, 2, or a 3. That's basically what we want to do. That's the task, right? So we are going to do it in 2 different ways, okay? All right. So before, maybe I will not show the first way. Let's do the second one first and then come back to the first one. Okay. Okay. So I hope everyone's paying attention. So basically what I'm telling you now is that, when we write in, when using a pen, right? Each of us have our own, what do you call, handwriting patterns, right? So everything, even if all of us, write number 1 in a piece of paper and then compare, right? Everything will look differently somewhere or the other. But then we all will know, though your way of writing 1 is different from my way of writing, if you write 1 in your own handwriting, I can identify that as 1, right? No problem there, right? So that we are trained, our brain is trained to do that kind of, recognize that kind of patterns. So now, let's say that I'll tell you that all these are number 1. Yeah, I have 10 images. This is 1 image, second image, third image, fourth image, and so on. All of them corresponds to number 1. Let's assume that these are written by different people or different fonts, however it is. Okay. And then I show you this and then say all these correspond to number 2. And then this one, I say all these images correspond to number 3. Okay. Right. So everyone just look at it for like 10 seconds and then, we'll move to the next slide. Okay. So now let me open the chat. So let's see, what is this number? What is this number? Is it 1, 2, or 3? That's 1. Okay. Thank you. And how about the second one? Number 3. And then the third one should be number 2. Right? So what is it? I mean, this seems like a simple exercise. Why are we even doing this, right? I'll tell you the relevance. I will now go back to the first method, right? And then say, okay, so let's go back to this only now, right? So now if I tell you this, and then tell that if you know programming, right? If I tell you, you should ask your computer to do this now, right? This is how we process, right? Because we have seen these numbers, we have seen these images, and we have found some patterns, right? And then based on these patterns, we answered the question, no problem, right? So now if I ask you, let's say I write a code, computer program on a computer to make the computer do this thing, right? So what we would do, we would do something like this. What we would do is, we will say, we'll make rules saying if there is only one horizontal line, that means it's a 1, right? And then we'll say if there are 2 horizontal lines and then the top line is smaller than the bottom line, it's 2. And then whatever rules we learned for the third one, right? So this is exactly what we would do on a computer, saying, okay, look at it and then see which one is it, right? Which, based on the rules, we make some rules and then say based on that you identify, right? These rules are pretty straightforward, right? When you look at these rules, you must be able to identify also, right? That's the second approach. Okay. So now what is the difference between the two? So the difference between the two is that, in one case, we have to tell someone the rules or the conditions based on which an answer is obtained. In the second, or the other approach, right, what we gave is only few examples. We said, these are the examples that you have, right? You learn yourself. I'm not going to give you any rules, right? You learn yourself how to identify 1, 2, and 3. I'm just giving you examples. Okay, I'm showing a lot of images and saying, okay, these are the images corresponding to number 1, 2, and 3, and then we have learned ourselves, right? Nobody told us any rules. That's exactly the difference between traditional computing and machine learning. Okay, so what do we mean by that? Let's go to the next slide. Okay, right, what is that's what I was trying to explain. What is the difference between the first, the rule-based one, and the self-learning, or the one which we learned ourselves? That's the usually the traditional computing, and the second one is what is the machine learning. Okay, so what is traditional computation? The traditional computation is that you have a program code, right? If some of you are already doing drug design, or somebody, I do not know what is planned for the rest of, I don't know if you're going to do some exercises and all those things, but you usually have a program, right? You have a program like, for example, Doc or Autodoc or Schrodinger, or there are several such programs that are available, right? So that could be your program, and then you have an input. Okay, you take these two and then use a computer to get an output. Okay, that's what you normally do. So that's what we do. So for example, I take a program called Autodoc, right? That's my program. And then I take protein structures and ligand structures as my input. What does the computer do? It takes these two structures and then uses this program to dock the ligand to the proteins, and then gives you a binding affinity, right? Or a docking score, right? So that's how we normally do. So when it comes to machine learning, for doing exact same thing, now this is very, very different. What is different? What we have done, as you can see, we have swapped the program and the output. When you go from here, the program is on the left, output is on the right. Here, the output is in the left and program is on the right. So basically, you give the inputs and then the corresponding outputs, right, to a computer. And then you ask the computer to learn a program that will take this input and produces that output. Okay. So that's basically the idea of this. So once you have this program, right, and then I can do this as usual, right? So originally without this, right? So you give examples like what we did, examples of images and corresponding answers. So we give inputs and the corresponding outputs. We ask the computer to give me a program or a model which will take this input and produce that output. So now what I can do for the future inputs, I can take the future inputs and then start predicting the outputs. Okay, that's the fundamental difference between traditional computation and machine learning. Okay, right? So that's what I said. So, you know, instead of taking a program and a protein ligand structures, you take the binding affinities. It could come from theory or it could come from experiments, and then the corresponding structures, and then tell the computer saying, for this set of proteins and ligands, these are the binding affinities. Okay, so you try to learn the relationship between the two so that I can use it for the future. So that's the idea. Okay. All right, so let's take an example. Maybe, you know, we'll take another example. Anyway, I think we can go up to 11:15, 11:20. I think so. We will take you know, another example here. Okay. So let us say I will come back to this slide. Just give me a second. Okay. So let us say I give you values of, let's see, for 2, a value of 4, 50, the value is 101. Let's say for 25, the value is 52. And for, let's say, value of 15, the value is 29. Of 8, it's 8, 100, it's 2, 100, 501 is 1000, and so on. Okay, so let us say, you know, let's call this X and this as Y, right? Okay, so let us call this as X and this as Y. Okay, so now, you know, I'm asking you, let's ask a question saying, okay, so in the same sense, for a value of, let's say, 75, what is the value of Y? Can you find out what is the value of Y for a given value of X equals 75? Okay, 149, someone says 150, 148, 151. Okay. I mean, you are allowed to give only one value, let's say. Or someone 153, 150. $152. Okay, so, you know, all right, so, you know, basically you are somewhere in the range of 1 plus 150 plus or minus 1 or 2, something like that is what I'm getting there, right? Okay, so how did we get this? You know, just based on, you know, these two series, you know, again, like the approach number 2, we did not need a formula. I did not tell you how an X and Y are related, right? Nowhere I showed that Y equals to X. This is not something I did not give anywhere, right? I did not tell you this thing. What all I gave some examples, right? These are the inputs, these are the outputs. What is that you have done for telling this? You have tried to figure out what the relationship between the two is, right? For example, someone says Y equals mx plus c or something like that, right? Okay, so basically what you have done is to figure out the relationship between X and Y, right? This is how our brain processes things. Okay, again, when you have to ask a computer to do it, usually what do? We say that this is the relationship, right? So once someone gives you a value of X, use this relationship to calculate the value of Y. That's how most of the computer programs are written. Huh? So that's what we do, right? But then in the other case, what we did is to figure out the or learn that the relationship between X and Y. Okay, let's go back to this slide now. So in machine learning, a basic machine learning framework is this. Given a set of inputs X, okay, let's assume I know a set of values of Y. I know X, Y pairs, okay? What is that I want to do? I want to figure out what this function F is. Okay, that's exactly what we did here, right? So I gave a series of X and then the corresponding values of Y, and then we figured out what the function is. Function is, 2X is the function, right? So that's basically what we want to predict. That is, in principle, the machine learning framework. So given a set of inputs and the corresponding outputs, the computer has to figure out what is the function that, will take X and give you the corresponding Y, right? So again, these two words will be used a lot in the, machine learning frameworks, where you do a training first. What is a training? Given a large number of XY pairs, that is the X and Y combinations, estimate F. This is what we want. Estimate F. How do we do it? Usually by minimizing the difference between the predicted Y and the actual Y. Okay. So that's, it could be, root mean square deviation or mean absolute error, different kinds of things, or, you can have different kinds of loss functions. But anyway, that's not what our interest is right now. Okay. So what we want to do basically is that I'm going to estimate an F. Okay. The process by which I estimate F for, given value of X, you get Y, right? So that process is what is called training. Okay. So, and then in this process, we minimize the difference between the predicted output or the predicted value of the output and the ground truth or the actual value of the output. And what is the testing? Now we use this F now, right? Once the training is done, I have F. Now I take the F, and then for known examples, I take the values of Y and then predict Y and then see what the actual difference is. So for a new value of X, use F coming from here to predict the value of Y and then look at the difference. Okay, that's basically what is a testing, right? So that's it. So, that's the, I think we're taking a longer, so we will skip some slides possibly. Okay, so that in principle is the, or the machine learning, a basic, again, I'm not a specialist of machine learning again, but then, that's the basic framework of simple machine learning methods. So usually that F we saw, right? That has to be a function. Huh? So usually we have a artificial neural network there. Okay, so the neural network is like a universal approximation for any kind of function that exists. Okay, that's how most of us do it. Sometimes it may not work, but most times it does. Okay, so now let's go to the application part. Okay, so that's the third phase of the talk. So first, I try to give you some context of why artificial intelligence. Okay, where is it useful? What is the relevance of these methods in drug design? The second phase, I try to take some very simple examples and then try to explain what we mean by machine learning in very, very simple terms, and how that is different from the traditional ways of computing. Okay, right. So we go to the next, the third part, which is the applications, actual applications in drug design. Okay. Right. So artificial intelligence methods, or the AI machine learning methods, are not so new in, especially in the area of drug discovery or drug design, right? So people have been using it for a long time. Actually, this is like the artificial intelligence existed for a long time as an area, right? And then the recent surge in the trends and everything is due to possibly very high the hardware itself has improved quite a bit, and then the libraries that exist, and then the data sets that have become available, and of course the kind of hype, right? There's a lot of hype around this particular area, and so there's a lot of hype around this area, and that also contributes towards why there's a lot of activity in this particular space. Okay, so this is an example of or this is actually one of the first papers. You can see the year 1973, right? So where they used the perceptrons for calculating or predicting the pharmacological activity of a class of molecules called 1,3-dioxanes. Okay, so they take the molecular representation and predicted the pharmacological or the activity of that molecule using a perceptron network, right? They were reasonably successful, right? But then, sorry, one second. I don't know what I did. So this was reasonably, successful, right? But then, people did not, continue to, or this did not get the kind of attention that it is getting now, right? For example, in the last, in the last 3 months, right? I have given this particular talk, I don't know, maybe 6-7 times, right? There's so many other programs also have happened. There was also one other FDP from NIT Warangal, I think, where we there's a lot of activity around this area because of several other factors, right? But then people did continue to use this, but then very less number of people. But then in the last few years, it's kind of exploded, right? I should update this data, but its activity is like growing too much in this area. Okay, so now with respect to case studies, what all I wanted to say from this slide is it's not very new in terms of application of AI methods in drug design, right? People have been using it for a long time, right? Especially in the area of computational drug design, people were using like methods like SVM, random forest, linear regression, and things like that for even shallow neural networks. People have been continuously using it, right? Okay, right. So let's let's look at some case studies. Okay, we have how much time do we have, Professor Vidyavati?
Sir, Dr. Kumar?
Yes.
Pardon me?
How much time can I take more?
Yeah, you have, I think, another 45 minutes.
Okay. All right. So, okay. So let's say we have protein structure prediction. Is, as I said, this is not a work that we did. This was done by some other group. Okay. So what is what is protein structure prediction? Right. For that, we need to have a qualitative understanding of this. Right. So you have protein when it's synthesized, let's say, right? Let's say in a biological system, like in a cell or in a lab, wherever that is, right? So the protein slowly folds to, slowly means it's still in the order of seconds or sub-seconds. Yeah. So it folds to form a native structure or a unique three-dimensional structure, and this structure is necessary for it to perform its duties or its functions. Okay, so the structure, this it has to fall to this form before it can um, perform a function, right? So getting the sequence is easy, right? But getting a structure is not so difficult. Why is structure important? Again, go back to the original slide of the drug design, right? When we have to have a target structure for us, it's good to have a target structure for us to start making or designing molecules which will inhibit that particular structure. Okay, so how do we do this, right? One is experimental ways of doing it, for example, X-ray crystallography, and second is computational way of doing things, like for example homology modeling, comparative modeling, right? And other kind of methods, right? People have been developing, a lot of people, I mean, it's a very active area of research, and we have progressed a lot in the last few decades. People have been working for a long time now, and then we have progressed quite a bit in terms of taking a sequence and then predicting predicting the structure. Okay, right. Okay, so this is how the process works. Why does it fold? Why does it fold from here to here? It folds because let's say this is the hypothetical free energy surface. Okay, so let's say that's the free energy, or delta G, let's say. Yeah. Okay, and this point, the black point here, is the free energy of the structure that is shown here. Okay, so you can follow the changes happening here and the change, how this black dot is moving. Okay, it's a movie, so you can watch it for like 10 seconds maybe, right? So I hope you can see that the structure is slowly going from a completely disordered state to a nicely folded state, right? So originally you saw the structure was very, very different, right? Like, this was the unfolded state of the protein, and then the energy of this is somewhere here. Okay, but then during the folding, the energy slowly changes, and then the structure also changes, and then the energy decreases to go to a state which is very stable. And then you can see that the structure looks like actually it does look like a protein now. Okay, right. So this is basically a protein folding, right? This is a process which is very, very difficult to simulate, though there are we have progressed quite a bit, especially the small proteins we are able to fold, and there are a lot of papers that have looked at protein folding, and then they are able to fold the proteins, right? And then to go to correct three-dimensional structures. Okay, so what is the task? What do you mean by what is it we are trying to do? So computationally, what we want is, given a sequence of a protein, we want the structure of the protein. Okay, so I'm not going to go into the details of how this is done. I'm just showing you the results. Okay, so the result is, as you can see, the ground truth, the actual experimental structure is given in green, and then the predicted is given in blue, okay, for 3 different proteins. Okay, so this got published recently, and this particular method, okay, this was actually a competition called CASP, right? And this particular method predicted 25 out of 43 structures accurately, and then the runner-up predicted 3 out of 43, right, with respect to a certain measure. Okay, so this is just these numbers alone do not capture the actual status of the competition results, but yeah, but I mean, just it is actually unfair to just show these two numbers and say that the winner predicted so much higher than this, but yeah, but then maybe this one gives a better scenario. So this basically means this is with respect to over the years of the competition. Okay, so this is a recent one, happened in 2018 end, I think. Okay, so with respect to years, right, we have slowly moving towards predicting it correctly. 100 means perfect prediction. Okay, so you're slowly moving towards higher accuracy in terms of predicting 3-dimensional structures of protein. Okay, so we are moving forward, and then there is a sudden jump that is possibly because of the kind of AI methods that this group used. Okay, so and then this group actually is what is called company called DeepMind, which is owned by Google, if I'm right. Okay, so you can see you can see the number of entities or new entities that are coming in this application of AI for drug design space, right? So this is a company owned by Google, and they are doing protein structure prediction, and they have done developed a lot of algorithms for disease diagnostics, right? And there is a paper from a research group in Facebook a couple of years ago where one of my students also works. He's also part of the paper, where they took the protein sequences and trying to understand some disease conditions, right? So recently, and then I can keep on giving many more examples of how a traditional internet company, like a social media company gets into this kind of space. That's how much the interest is, right? So let's second case study, or what is it that I wanted to tell here, or what did we try to see? That the application of, or the or the extra effort in terms put into the engineering aspect of AI methods applied to protein structure prediction has increased our accuracy in terms of predicting structures of proteins accurately, significantly. Right. So I'm going to show these examples. Maybe yeah, let's take this one, and then maybe we'll skip and go to this. We'll see. All right. So let's now molecular property prediction. Right. What is molecular property prediction? Basically, it's like I have a molecule. Right. We always ask. I have a molecule. What is its property? Right. This is a question that we always ask. Right? For example, if I have a molecule, what is the energy of the molecule? What is the, let's say, dipole moment? What is its spectra? Okay, so, and so on, right? So you have a large number of molecular properties associated with every molecule, and I want to calculate properties. How do we normally do in traditional computation? We use computational methods like quantum mechanics or density functional theory or molecular dynamics simulations and so on, right? So these are some of the popular methods that we use to calculate properties of any given molecule. This is what we normally do, right? Okay, so all right, so first example that I'm going to show is the prediction of molecular energies. Okay, All right, so again, let's skip a couple slides and go here. Okay. So this we published recently. So this is a work from our lab. Okay, so let's say I want to predict the energy of molecules. Okay, so let us assume that what I want is to calculate energies of molecules. Okay, what do we normally do? What we should be doing? We should be using quantum mechanical methods to calculate reliable energies of molecules, QM methods, or let's say density function theory methods, both of them. Yeah, so that's what ab initio or DFT methods. I want to calculate energies of molecules. Okay, so if the energies, if excuse me, if the size of the molecule, right, is small, right, then this is possible. If the size actually increases, right, so actually no, this is add 1 or 0 to this. I should add it next time. So basically, if the size of the molecule increases, then we cannot use quantum mechanics as a method, because I was going to give a couple slides background on this, but then we will not worry about it. Let's just for now take it as like quantum mechanical methods are computationally very, very expensive, right? So we cannot so we cannot apply quantum mechanical methods, for example, for protein systems. Okay, so what do we do when the number of molecules, or when the size of the system becomes larger and larger? We use approximations like a molecular mechanics method. Okay, so for example, in case of drug design for docking, for example, we use scoring functions, which are kind of which have equations similar to these plus some more terms added to them and things like that, right? Okay, so but then what is the problem with this? In terms of accuracy, these are not so great compared to quantum mechanical methods. So what do we do? So but then we cannot, in practically, what do? We have to use this. Okay, but now the idea that again a large number of groups are working on is to see if we can come up with a machine learning based potential energy function or a machine learning based force field, right, which is as expensive as molecular mechanics. It's not so very time consuming, or it is computationally still not so difficult like molecular mechanics, but then in terms of accuracy, it's more accurate than molecular mechanics. That's the whole idea. Okay, so this is the work that we did. For example, for any given small drug-like molecule, small molecule, what we want to do is predict energies. Okay, so one of the things I did not tell is that we talked about X, Y, and then the need for predicting F to connect these two, right? So this is what we talked about, right? So that's what we said, machine learning, because I'm trying to learn this function. Okay, so one thing I did not tell, X need not be your molecule, right? X could be something that is derived from molecules, right? So you have a molecule, right? And then instead of how do I represent the molecule in terms of X? Okay, so that's basically this particular slide. So I'm representing a molecule in terms of its bonds, angles, non-bonds, and dihedrals. We'll not go into the details of this. It's just that one way, or we came up with this way of representing molecules for doing this exercise. I have a molecule. I derive some features from molecule. So these features represent this molecule, and those features are used to predict the energy of a molecule, or first used to get the or train the model and then predict the energy of the molecules. Okay, so this is what we do. So you start from the Cartesian coordinates and then the atomic numbers of the molecules of the atoms in a given molecule. For example, this one, you have like atomic numbers of each molecule here, and then the position, the three-dimensional position of each molecule. And then based on that, you generate or you derive features, right? The X's, right? And then you use artificial neural network to, so to predict the energy of the system E. Okay. So again, in terms of the discussions that we had before, this is the molecule, this is X, this is Y, and the in-between part, that's basically my function F. Okay. So based on known examples of X and Y, I learn what F is. Okay. So once I have learned F, right, for a set of molecules, and then I can go use this F to predict energies for any future molecule that we have. Okay, so that's basically the idea. So I'm just giving you some examples of or some results. What is given here is a set of molecules which are C11H22 isomers. You know, these are randomly chosen and made sure that they are reasonably diverse. Okay, and that's what is plotted is the relative energy of these molecules. Okay, so that's the ground truth or the results from density functional theory calculations. Okay, and that is the machine learning model. That's the results from the machine learning model, that, you know, the energy is predicted by the work, but by our algorithm is very, very close to the DFT energy. Okay, just to give a comparison, this is a set of this is another method which is a semi-empirical method which takes much longer in terms of time to calculate the energies, and that is something like this. Okay, so we do pretty good. So the AI methods do pretty well. So again, you know, different comparisons in terms of calculating or in terms of learning the potential energy surface, right? For example, this is the energy corresponding to stretch between a bond. You know, that's the energy change, right? And you know, you can see the DFT and then the ML1 both are reasonably good again here, and so on, right? This is for bending, and then these are comparisons of some reaction energies. These are the ground truth. That's the reality, let's say. And the blue ones are the machine learning based models. Okay, and then the yellow ones are the semi-empirical ones. You know, it even gets the order incorrectly sometimes, right? Okay, so that's another example, second example of how one can use machine learning to do property prediction, right? Okay, I will not go into the details. So we also have other examples where, for example, we predict free energy of solvation, right, which is useful for calculating some pharmacokinetic properties. For example, that's the experimental free energies of solvation. That's the predicted free energies of solvation, predicted based on a machine learning method, and then we do very, very well in terms of the agreement between the two, right? So we'll skip that topic and then go to this. That's the last topic that I wanted to discuss. Maybe I'll take another 10 minutes. Again, if there are any questions, please feel free to intervene. Okay. All right. So most of the times when we do drug design or do any experiment or anything in chemistry or medicinal chemistry or pharmacy kind of exercises, okay, so what we do is this: we take a molecule, right? We take a molecule and then and then predict what is the property of that molecule, right? That's what we do. Like, for example, here, for a given molecule, what is its property? Right? That's what we just saw also, right? Whether it's a computation or experiment, it doesn't matter. right? So this is what we do. So why do we do this? We do this because we want to do we want to do this. So you want to go back and then try to understand the relationship between, right, structure and property. So for example, in drug discovery, we talk about structure activity relationship, quantitative structure activity relationship, for example, right? So we start with some initial guess molecules, we calculate properties, we try to understand the relationship between the two, and then try to come up with new molecules that are better and better in terms of the property, right? For example, I say that I want a molecule which will bind to a certain protein. Okay, so let's assume that's my question. Okay, that's my research question, right? So my objective is to design molecules that will bind to a certain protein strongly, right, which is a high binding affinity. Okay, so that's what we want to do. But then what we do is exactly the reverse, that we actually take the molecule and then calculate its properties, which is kind of not so efficient, but that's what we do, right? So this is a question we ask. We say, I want a molecule, right? I want a molecule with a certain molecular property. That's the question that we ask. Okay, so that's where this particular figure is from this paper, right, where they proposed reinforcement learning to do to solve that problem, where they said, instead of taking a molecule and predicting the property, you ask an algorithm saying, I want a molecule with a certain property, and the algorithm will generate molecules for you, right? So that's the generative model, like a model that can suggest new structures. Okay, so there are two machine learning models now. Okay, so this is the first one. This is the first one, which is called a generator, and a second one which is called a predictor. Okay, so I have two networks here, let's say, for now, right? It's a whole, all integrated in one thing, but let's assume that there are two for now. Okay, so one is called a generator, second is called a predictor. Okay, so what does this generator do? It generates new molecules, right? It just keeps giving you new molecules. So predictor model is pre-trained, like the one which we saw before, where you have the energies, right? So you take these molecules and predict that particular property. And then if the property that is predicted is close to what we want, right? If it is close to what we want, then you give a reward to this particular generative model, right? If it is not close to what you want, you penalize that model, right? So, and then when you maximize the reward, or you say that the reward has to be maximized, then slowly the generative model will start suggesting molecules which have properties that are something that we want, right? You can the simple way to understand this is like this. Let's say we get one of us, or I don't know how many of you have dogs in house, right? So initially, how do we train a dog? We say, okay, we ask the dog to do something. Let's ask the dog to stand, right? And then if the dog stands, right, what do? We give a treat, right? If he doesn't do it, we are not happy. We show that we are not happy, right? So basically, dog likes treats, or likes the biscuits or whatever, right? So it will slowly start learning to understand what you are saying and then do things accordingly, right? It's very, very similar, right? You have a generator which starts generating molecules. There is a model to check whether the molecules are something that is useful for me or not. If they are useful, you give a treat or reward. If they are not useful, you penalize it. And slowly, when with respect to certain iterations, the generator will learn that it is getting rewards for a certain kind of molecules, and then it will start generating that kind of molecules. Okay, so we will, I will demonstrate this in, with respect to two properties. One is log P. Many of you may know, log P is the partition coefficient, which helps us assess whether a drug molecule can, cross the lipid bilayer or not. Okay, so it has to have a certain value for it to be able to cross the lipid bilayer, right? Because you have a very hydrophobic environment, and this is all hydrophilic, so it has to have a balance of hydrophilic and hydrophobic properties for it to cross the membrane. Second is when we propose new molecules, right? Like for example, let's say I propose this. Someone should be able to make it, right? It should be possible to synthesize in a lab. Okay, so there is something called a synthetic accessibility score. Okay, so which assesses how difficult it is, to synthesize that particular molecule or not. If the value is very high, that means it's very, very difficult to synthesize. If it is very low, then it is easy to synthesize. So now let's apply this to these two properties. Okay, so what we are trying to say now is, let's just look at this plot for now. Okay, so I'm saying my log P value, the log P value has to be in this range for it to enter a cell. If it is beyond this or below this, it cannot enter the cell, and hence those molecules are not so very useful. Okay, and come to this one now, right? So now I say, with respect to the synthetic accessibility score, I want to want the numbers to be as small as possible because it's easy to synthesize. I don't want molecules which are impossible to synthesize or very, very hard to synthesize, right? I want molecules which are easy to synthesize. So if we do not do any training or if we do not bias the system, so this is how the distributions look like. Once I use the reinforcement learning, right, you can see that slowly the distribution is coming towards or between these 2 points, saying almost 100% of the molecules that are being generated or in this range. That means, almost all of them can actually enter the cell. So I don't have to deal with molecules which are away from this region. And similarly, in this one, slowly the algorithm starts generating molecules where almost all the molecules that are being generated are fairly easy to synthesize, and I don't have to deal with molecules which are hard to synthesize. Okay, so that's, you can apply this to many other properties. For example, we looked at log P, benzene, number of benzene rings, melting point of the molecule, and different things. Okay, so with that, I think we should stop. We, just to quickly summarize, modern machine learning methods, are fundamentally changing the way we perform, computations in drug design. Okay, so it is becoming, it is like a disruptive technology where it has started contributing to in different ways to the area of drug design, right? And then the as I showed some examples, right? AI/ML methods, they have a great potential to make the whole process of drug discovery or development process very efficient. Again, having said that, we are in the initial stages. We just have started in some sense in the last 2 years, right? Very seriously in this space. There's a lot more to do, and progress in this direction will make things very, very efficient. Okay, so with that, thank you. And if there are any questions, we can take or stop.
Yeah. Thank you. Kumar. Thank you very much. Pooja, are you there, Pooja?
Yes, ma'am.
Can you coordinate some of the questions if
Yes,
there are
ma'am.
any in chat box?
As of now, there are no questions in the chat box. Participants can start posting their questions in the chat box.
Now the session is open for discussion and for clarification of any doubts.
Madam, can I ask?
Yes, yes, you can
Yeah.
ask. If you have,
Yeah.
if you are able to unmute, you can ask all the participants. Otherwise, you can post in chat box.
Yeah, ma'am. May I take the question?
Somebody is
Sir,
already asked. Wait
Yes.
for a minute, Jeevan. Okay. Shaheen?
yes,
Dr.
madam.
Yeah, you carry on.
Sir, you have said in the first slides itself that Bayesian machine learning is very useful in drug target identification. Is it what is the meaning? Means a novel target we can identify, or the whatever the, for example, if you take cancer, we have so many targets. Whatever the available targets, we will be able to target. We will to identify any one which is more relevant, or it will be whether it will be totally novel.
Yeah, so that's the good question. Again, our work in this particular area of drug target identification is very small. Actually, we have been working, especially on the COVID-19 one. We are working on some aspects of it, but the idea is that there are I cannot answer specifically, but then. The idea is that when we analyze, how do we, for example, one of the ways to identify targets is to look at the biological pathways, right? So, for example, let's say viral infection. So you have a certain number of proteins from the virus, you have a certain number of proteins from the host, and then a large number of these proteins interact within the viral proteins, within the host proteins, and then among themselves, right? And then we do some kind of mathematical modeling, for example, and then we can identify targets. That's, that is very, very efficient, and we have been using it for a long time, right? So now, with the machine learning, there are some papers, for example, not only these protein-protein interaction networks, you can also use all the information about all the drugs, how the drugs interact with other proteins, right? So you're able to combine information from different scales, and you are able to identify new targets that we have not really identified before, right? So I think that's exactly what your question. So are we identifying the same targets or identifying new novel targets, right? So in that sense, yes, we can use these new methods to confirm or that's one way of identifying targets, and that can identify things that could have been identified by others as well. But in addition to that, because computers can, or these algorithms can see things in a much more larger dimension than we can, or than traditional computations can, they do suggest new novel targets which we do not know. So it's so that way it's more efficient. I hope
Thank you,
I
sir.
answered. Yeah.
Thank you.
Thank you. Thanks.
Shaheen, may I take a question?
Yes, Dr. Jeevan. Yeah.
Dr. Deva, how are you? You were very super senior to me in Shastri sir lab when you left.
I saw your name, and I
Yeah.
Yes,
You are doing
a few, but I did not think it was you who is.
Yeah, okay. You're doing extremely good in ML, and we are also doing here. Now we have generated one model with linear regression, only taking around 5,000 molecules in EGFR. But a little bit we are undirectional in terms of predictability of new compounds. So could you help us in that angle?
Yeah, I mean, sure. I mean, we have, of course. Please write to me, and please send an email. I think it's on YouTube, or else I would give you my phone number right now. But
yeah, just drop me your mobile number so that I can catch you.
Yes.
Okay. Thank you.
Please mute Shaheen or somebody else. Please mute your audio. There is a disturbance.
So there's a question from Sujit Das. How machine learning will help in finding efficacy of a new synthetic or new natural isolated drug?
Yes, so see, though when we hear of how disruptive these new technologies can be, and how we talk very nice things, right? But when it comes to reality, right, all these things are limited by the availability of data. Okay, so in that sense, if you are looking for calculating, for example, efficacy of a given compound towards a given target, as of today, there are no algorithms that are available that will let you do it, right? There are some, like there are some algorithms or some methods based on machine learning which will give you a better answer than the traditional scoring functions. Okay, but the reality is it's still not good enough, right? That's what I said. So we are just beginning in this area in terms of how we can take all these processes to a next level in terms of more efficient and more accuracy, right? That's what we can do at this point, right? It's not like 2 years ago that these things were not there. Now that AI is there, it should solve all our problems, right? So all these questions, it's a very good question and is very, very relevant. Can we find can we find the efficacy of any given molecule towards any given target? Yes and no. Algorithms can do it, but I do not think we have the kind of data that is needed to do that, right? So we are limited by the data, but then we are improving quite fast, quite aggressively in that direction. Today, no, but soon, yes. I should not say no. It's partially yes, because we can do better than the traditional docking scores based on the existing ML-based methods.
Yeah. Pooja, are there any more?
That's it, ma'am. I've given an option to participants also to unmute yourself and ask any questions.
Yeah, definitely.
So if any of you cannot access the chat box.
So I'm just typing my email address. So I don't want to put my phone number here, but if Ravindra, if you can drop me an email, maybe we can have a chat. I'll share it.
Yeah.
Others also, I mean, please feel to write or anything. So we are, we collaborate with a lot of people, right? And then we also train a lot of people. So.
Thank you, sir. Is there anybody? If anybody is interested to ask any doubts, we have another 5 minutes only. Yeah, if not, let me take the opportunity to thank Dr. Kumar. Thank you very much, Dr. Priya Kumar, for your elaborative lecture on artificial intelligence and machine learning methods for drug design. Also for making all the participants active by giving tasks to the audience. The AI and ML methods are becoming very potential in the area of molecular sciences, especially very effective in drug design, prediction of active molecules, and also to find the feasible methods to synthesize those molecules. Dr. Kumar highlighted very well about all these with successful examples, research publications, and from basics with the 3D pictures to understand easily, even by anybody. Thanks a lot. Thank you very much, Dr. Kumar. And also thank you for accepting our invitation and spending valuable time with us for enlightening all the participants of this FDB. Thank you very much.
Yeah, thank you. Thanks. It was nice.
Now, I request all the participants take another 10 minutes break for the next session. The next session will be started shortly by 11:45 by Professor A. Uma Maheshwari Devi on in silico strategies in drug discovery. Thank you. Shall we give break, Sridevi? Yeah, I have announced also.
Yes. Okay.
Thank you, Sridevi.
Okay. Thank you, sir. Devapriya Kumar, sir. Thank you very much.
Oh, you're welcome. Yeah, so I was wondering.
Thank you. You are given very informative talk, and it is very new to us, and how the machine learning is helpful in the drug design. So we are very thankful for that, and we expect cooperation in future also, sir. Thank you.
Yes,
Thank you
yes,
very
yes.
much.
No problem. Bye bye. Yeah. Bye everyone.
Bye.
Take care.
Bye, sir.
Take
Bye.
care.
So by sharp 11:45, we will start the session, second session. Okay.
Dr. Uma Maheshwari,
Uma
has
Maheshwari
come? She has
madam
come?
will.
Has she come or has she joined?
I will check.
Yeah.
Madam has joined. Uma, good morning.
Yeah.
Hi, Sridevi. Am I audible?
Yeah, yeah.
Okay.
Yeah. Sridevi, you have to unmute, it seems. Yeah. See, Uma Maheshwari, this is Professor Vidyavati. You are audible and waiting for your talk. Yeah.
Okay.
How are you?
Good. Fine.
Okay,
Meanwhile,
within 10
shall I share my screen?
Okay.
Yeah.
Okay.
We have another 10 minutes. We asked to give.
Okay,
we
okay,
asked the
okay.
participants to join by shortly
Okay.
11:45.
Okay.
Right.
Okay.
Before sharing your slides, we have to give your biodata, no, Uma? For that,
I
Pooja
already
has to
given to
share.
you.
Huh?
Yeah, that we want to share that slide. Uma, just wait.
Okay.
Yeah, okay. Right.
Okay.
Is Shahin ready?
Shahin is there.
Yeah. Okay.
So within 5 minutes, we'll start. Okay.
Okay.
Hello. After a long time again.
I think I am visible now.
Yeah, visible
Yes.
and audible.
Okay.
Yeah.
Visible and audible also, both.
Yeah, total FTP, yours only. Your subject, total FTP.
Yes.
Hello, madam. Good morning, madam.
Yeah.
Shahin, how are you?
Fine, madam.
What about your students? Uma, are they coming?
Final year students, they completed their exams.
Yeah.
So the course is completed. And now second semester students, they will come from 10th onwards.
Okay.
So in the month end, they will be having exams.
Okay. Do you have any COVID duties in your,
No.
premises? There are no.
No.
There won't.
It is only for doctors.
Doctors. But based on the availability of the beds and space, they are spreading to other beside rooms or campus also.
Now there is a reduction in the number
Yes.
also. There is
Now.
no problem. Previously, there is a big demand for that beds.
Yes.
You may go for tea or something, some break.
Yes.
You are sitting from the morning itself.
I was moderator for the first previous session.
Yeah.
Just I'll leave. When we start, we can do.
Okay.
No problem.
After 5 minutes, I will join. Otherwise, I will stop my video and audio.
Yes. No problem.
విద్యా మేడం, నేను మీకు పంపించాను చూడండి. టైటిల్స్ కెమిస్ట్రీ వాళ్ళవి రెడీగా లేవు, పంపిస్తాను నేను.
ఓకే, ఇట్స్ ఓకే, నో ప్రాబ్లం. Okay, Shaheen, I think you can coordinate now onwards. I'm leaving. I'm leaving in the sense I'll be here
Thank
only.
you.
Yeah. Madam.
Yeah. Okay,
Thank
bye.
you, Vidya. Thank you very much.
Yeah, it's our duty, no problem. Don't say thank you to all the moderators. It is our university and our department. Okay, right.
How are you, Uma? Classes are started for you?
మేడం ఒక 5 minutes break కి వెళ్తానన్నారు మేడం.
Okay, no problem then.
11:45 she will join exactly.
Okay. ఎందుకంటే some of the participants are also left. Okay.
Exactly 11:45 కి they will join madam.
Okay, we'll start also. Okay.
Let her join. మేడం జాయిన్ అయితే వెంటనే 11:45 కి మనం స్టార్ట్ చేసేద్దాం మేడం.
ఓకే, మేడం జాయిన్ అవగానే చేసేద్దాం. ఓకే,
మిగిలిన పార్టిసిపెంట్స్ యాడ్ అవుతా ఉంటుంది.
అవును.
గుడ్ మార్నింగ్ ఆరిఫా. Madam, we will start within 2 minutes, madam. Now
Okay.
participants are slowly joining.
Okay, okay, no problem.
Shaheen, you can start. Otherwise, it may be delayed. Okay.
Okay, madam. Okay, madam.
Hmm.
Okay. So, good afternoon, everyone. Good afternoon, and thank you so much for being this morning with us. So, I welcome all the participants and resource person. Today, I am very much pleased to welcome Dr. A. Uma Maheshwari Garu, a very special guest from SWIMS, Tirupati. SWIMS stands for Sri Venkateshwara Institute of Medical Sciences. So, Madam is currently working as Associate Professor in the Department of Bioinformatics, SWIMS University. So, overall, Madam has 23 years of teaching experience, and she has successfully completed 7 PhD students. She has successfully guided 7 PhD students, and so many are now working under her. Madam is involved in so many different extracurricular and co-curricular activities. At her department, she is involved in different professional activities. She has been doing as BOS person for different courses, for PhD courses, and we can see from her biodata that she has been a coordinator for BIF and a women grievances member. And the best part from her biodata is she has handled over 11 projects worth nearly 1 crore, nearly. So she has been a reviewer for very well-known journals. Natural science reports, computers and medicine and biology. Madam has around 90 international and national level publications in very well-known journals. And it's our honor, madam, that you are with us today. And Madam will be sharing her expert opinion on in silico strategies and their importance in drug design. And I request everyone to give full attention and to the lecture. And with this, I welcome Dr. Amineni Uma Maheshwari Garu to this session. Over to you, madam.
Okay. Om Namo Venkateshaya. Thank you, Shaheen, for your wonderful and nice introduction. And small correction in that, my title is not whatever you said. So Sridevi may be knowing. So my title is Omics to Drugs.
That's the later madam has changed. Okay.
Okay, madam.
Okay.
Hmm.
Okay. And greetings to all from my institute, Sri Venkateshwara Institute of Medical Sciences. And I thank this FDP program for inviting me. And a special thanks to Professor Sridevi for inviting me. Shall I share my presentation?
Okay, ma'am. You can.
Okay. Is it visible? My slides are visible?
Yes, madam.
Okay, okay. So today I will discuss about omics in omics to drug design by using artificial intelligence. So before going to this, I will give brief introduction about artificial intelligence. So all of you know that we are having lot of neurons in the brain. So with this only, 1 second please. So with this neurons, it forms into a network. It is called as a neural network. With the help of these neural networks itself, we can sense, reason, and think. So in the year 1950, the scientists, they designed neural networks artificially to mimic like a human being. So then it is named as artificial neural networks. So in this, artificial intelligence means whatever the thing which is mimicking like a human brain is called as a artificial intelligence. So this artificial intelligence, previously it was like a general artificial intelligence in the year 1950 because they used to develop robotics. And now with the advancement in the computational power, it was narrowed and it is named as narrow artificial intelligence. So this is because to give the narrow tasks or it focuses specific tasks. So in this day-to-day life, this artificial intelligence is a part and parcel of life, like our smartphones. To open the smartphone by recognizing our faces or recognizing our thumb or with the voice, we can open the smartphone. So all these things are possible with the artificial intelligence. So like that, it enters into our lives. So mainly coming to our problem. So this pharma industry, all of you know that there is this drug designing is very expensive and time consuming. And at the same time, it is having 90% failures because if 10 compounds are reaching to the clinical trials, only one is successful, means it is having 90% failures. And at the same time, it is having it is involving billions of dollars. So these scientists, they thought to overcome this problem, they introduced this artificial intelligence into the pharmacy. And in this decade, there is a tremendous change in the pharmacy industry. So this artificial intelligence is making computers smarter. How it is possible means by using machine learning. Means we are making the machines to learn the things. So coming to this artificial intelligence, mainly machine learning and deep learning algorithms are useful for our studies. Means mainly we are focusing on drug designing studies. So in this machine learning, there are 3 different types. They are supervised learning, unsupervised learning, and reinforcement learning. So whatever the data we are giving, it will take as input and it will search for the patterns and then it will classify and it will give the results. So sometimes it is called as a automation. So in this supervised learning, if you see the supervised learning, again, there are 2 different types. One is classification and another one is regression. Classification means suppose if you are having a breast cancer patient. So what is the size? Means we will be having some numbers, means it is a structured data we will be having. So it is having the different size and the age of the patient, like that we will be having some parameters. So based on that parameters, whether it is, the patient is having the disease or not, like that we can diagnose the disease by using supervised learning by applying the classification model. So regression model means all of you know about the drug molecule. Efficacy will be calculated in the form of IC50 values. So if we know the IC50 values of the some compounds, by generating the regression model, regression analysis, we can predict to our compound. So this is also in the form of a structured data. So numbers will be present. So we can predict this IC50 values by using regression model. So second one is the unsupervised learning. Previous one, there is a supervision is necessary to check whether it is correct or not. Means we are having the figures, so we can supervise. Somebody can supervise and do. So second one is the unsupervised learning. Unsupervised means, suppose if there is a disease like breast cancer diseases, there are different subgroups are present. I will show in the coming slides. So in this, different types will be present. There is no numbers are available for these particular ones. So there is only image will be present. In one area there is a densely present, and another area it is very light. So based on that, we need to predict what is the subtype of cancers. So this is possible by clustering that particular one. So here we are not having the structured data. If we are having the unstructured data, then we will go for the deep learning algorithms. So first one is supervised learning, mainly with the help of machine learning algorithms. These two are one and the same, but if there is a unstructured data is present, we will go for the deep learning. And this deep learning is more accurate than the machine learning things. So like that, we can cluster and we can give the subtypes, whether it is having the luminal type, luminal A, luminal B, like that also we can give for different cancers. And feature findings means we are having enormous data. So from that data, we can identify the drug targets by, it is also unstructured data. So we can do this one by using deep learning algorithms. The third part is the reinforcement learning. This is important because it gives you the decisions. So previously, suppose if we are doing docking studies, so we will take the compounds which are available in the databases. So if you want to take, if you want to get a patent, we cannot get a patent if it is the molecule existing in the databases. So we need to do the novel molecules. So once we are developing a novel molecules, again we need to do the docking analysis. And in this automation, it will generate a lot of conformations and it will add some functional groups like that. Automatically, it iterates and it will give the novel molecules. And also it will automatically compares with the existing molecules and it will give the scores. The top scored one will be given in the first rank. So like that, it will take the information and it will do lot of iterations and finally it will give the decisions. So this is called as the reinforcement learning. Here it will give the rewards because in this de novo synthesis, so whether this molecule is fitting into the active site or not, if it is fitting, then it will give the positive rewards. And also ADME properties also check whether it is having a good properties, then only it will give the rewards. So morning also sir told this rewards will be given in the reinforcement learning and so that we will get the output. So the same thing will be executed for experimental design. So mainly the difference is machine learning and the main thing in this is machine learning and deep learning. We are giving the data. From this data, we will get the output in the form of patterns. Okay. If we see this biological system, so ultimately we need to apply to our system. So our system is very complex in nature. So this system is having billions, trillions of cells, and each cell is having a genome. This genome is having 3 billion base pairs. Each cell is having 3 billion base pairs. So like that, we are having so many omes. So omes means genome, proteome, transcriptome, metabolome, interactome, and phenome. So this genome means the total set of genes present in a haploid set of genes present in a cell. So it will not change. But transcriptome means once the gene is expressed, it will be in the form of mRNA. So the genes, some genes are housekeeping genes, they are continuously expressing. Some genes, whenever there is a requirement, then only it will express. So this transcriptome, from the transcriptome, proteome will be present, and from the proteome, enzymes will be present, and these enzymes are involved in so many reactions so that it will form into a different molecules, like our small molecules, like biochemical molecules and hormones, signaling molecules. So with this interactions means with this signaling molecules, there is a cell-cell interactions, everything will happen with the signaling molecules. Once there is a change in the transcriptome, there is a change in the proteome, and if there is a change in the proteome, this reactants will also change, and finally interactions. The recent studies are showing, if you take the cancer patients, the same proteins may be present, but the interaction of the different proteins with the different groups causes cancers. So this is called as a interactome. So if automatically, if there is a change in the interaction, if there is a change in the protein, there will be certainly there is a change in the phenotype of the cell or organism. This best example is our butterfly. So in the this butterfly, there will be 4 stages: egg, larva, pupa, and adult. So in all the cases, genome is same. Only the difference is transcriptome, proteome, metabolome, interactome. So which causes the change in the phenome? Means physiological characteristics will be changing. So yeah, in our system, this each cell is like a industrial cluster. And if there is a health is good, these all are working together, and we will not have any problem. If there is any change in all these things, subtle change makes big difference. So this is the case in the cancers. So all these wombs are important. This right side, omics, means the study of these wombs is called as genomics, or proteomics means the study of all these things. So today I will focus mainly on the phenome and transcriptome. So the phenome, the drug target identification based on the phenome, drug target identification based on the transcriptome. So we will see the details. So this is the traditional drug discovery program. So traditional means it will start with the genome, target identification, target validation, drug discovery, lead optimization, preclinical and clinical trials. Later, it will enter into the market. So this is the traditional one. Now, by applying the artificial intelligence, previous one is a linear fashion. So here we are having the big data. Big data means it is not only huge in volume, at the same time it is also having diverse data. So this diverse data, like you have already seen these wombs. So this diverse data, the human being cannot analyze this. So we need computers. So this big data is available and computational speed is also available. So by integrating all these things, we can do the drug designing process. The previous one, traditional drug discovery, is a linear process, and this is the cyclic process. Why this cyclic process is required? Suppose if you are taking some cancer drugs, so for 1 year or 1.5 years, you will get resistance to that particular drug molecule. So what we need to do is we need to identify the novel molecules. So if it is in a cyclic fashion and already prepared ones are present in the database, so in the cyclic manner, you can develop a novel molecules. So the same thing here also, target identification, target validation, hit identification, hit to lead, and lead optimization, preclinical and clinical studies. And in case of if you are treating with the infectious diseases, these infectious diseases, they will develop some multi-drug resistance. So at that time also, these antibiotics will not work for that. So if we are having all the information about the genome of the organism and these available drug molecules, we can repurpose these drugs also, and we can give the new molecule, new drugs to the condition, based on the condition. So with this introduction, I will explain my work. So basically, I am working on breast cancers. So this breast cancer is a metabolic disease, and also it is endocrine and inflammatory disease, and it is polygenic in nature. So if you see this database is called as a GloboCam, means Global Cancer Registry. So it is having only updates up to 2018. That is why I kept this slide. And if you see this statistics, here this is the Indian statistics. Here also the breast cancer is the number 1. And if you take the globally also, the breast cancer is placing in the first position. And in case of mortality, it is in the 5th position. So it is somewhat curable. So before going to detail, we need to know the severity of the cancer by staging and grading. So here staging and grading will be given by the American Joint Committee on Cancers and Union for International Cancer Control. So they will be given in 3 terms, that is T, N and M. So if the T means this tumor, the extent of the primary tumor is called as T, and then N means the extent of cancer within the nearby lymph nodes, and M means metastatic condition. Here what happens is, once there is a cancer, these cells will proliferate. So this proliferation means the increased number of cells will be present at a given point. So it is mainly due to the proliferation of the cells, or regulation means stopping the apoptosis, means if there is no death, only proliferation also leads to the cancers. Once there is a cancer, once the mass is present in this particular site, then there is these cancer cells require more and more glucose and oxygen. So there will be need for the nourishment of the cells. For that reason, the system will develop the blood vessels to supply oxygen and also nutrients to these tumor cells. So this process is called as a angiogenesis. Once the angiogenesis is there, means once they are nourished, they will proliferate like anything. And once they are proliferated, these are in the form of a tissue. Tissue means all these cells are present intact with the help of ECMs, means extracellular matrix. So once these cells are grown, this tumor, it induces the matrix metalloproteinases. These proteinases, they act as a scissors, and they will cleave this extracellular matrix surrounding around the cell. So these scissors, they will cleave the extracellular matrix, and this cell becomes an independent cell. So once this cell is independent, it will already due to the angiogenesis, so many blood vessels are present in the tumor. So it will reach us to the blood vessel and invade into the blood circulation, means it will enter from the tissue to the systemic circulation. It will enter into the bloodstream. So it will reach us to some other distant part, and then based on the microenvironment, if the microenvironment is like soft, if the breast cancer means mainly it will go to the brain or it will go to the lung. Sometimes it will also go to the bone. So like that, it will choose the microenvironment wherever it is comfortable. Then they will stay there and they will spread. That condition is called as the metastatic condition. M0 means it was not spread to the distal sites. M1 means it was spread to the distal parts. And again, there is a grading, grade 1, grade 2 and grade 3. This grade 1, grade 2, grade 3 means whether it is having more spreading capacity. If grade 1 means it cannot spread very fast. So they are called as low grade tumors, intermediate tumors and high grade tumors. Means high grade tumors, they will spread very fast. So based on the site of the tumor, this cancers can be given some names. Like in the breast tissue, two types will be present. One is ductal one or in the lobules. So this breast tissue is having milk ducts and also lobules. So this cancer is occurring in the ductal region, then it is called as a ductal carcinoma. And if it is occurring in the lobules, then it is called as a lobular carcinoma. So again, there are two different types. One is in situ and another one is the invasive. In situ means where it is the original place, wherever the tissue, wherever the cancer is growing, it will grow in that particular area only. But invasive means it will move to some other parts also. And here, if it is in the duct and it is invasive, it is called as the invasive ductal carcinoma. Majority of the cases, 80% of the cases are invasive ductal carcinoma. Ductal carcinoma in situ means they will present in the ducts and they grow in that particular area only. Lobular again invasive and in situ, the same thing. If it is in the lobules or it is spreading to the breast tissue also, then it is called as the invasive lobular carcinoma. So another one is the inflammatory cancers. This inflammatory cancer means it is only 1% of the patients will get this inflammatory type of cancers. And the treatment will be chemotherapy, hormonal therapy, targeted therapy. So once there is a menopause, the cancers are related to lobular carcinoma in situ. And if they are taking some hormone replacement therapy, the patients will get invasive lobular carcinoma. So based on the molecular subtypes, based on the immunohistochemistry, these cancers are classified into luminal A, luminal B, triple negative, HER enriched, and normal like breast cancers. So in these three different markers, molecular markers will be present. One is ER means this estrogen, or PR means progesterone, estrogen positive or progesterone positive means these two are the hormones. Hormone positive or HER means EGFR. So HER positive. So if the three things are negative, then it is called as a triple negative cancers. HER enriched means here only EGFR enriched, overproduction of EGFR will be present. So luminal A, luminal B, the difference is both are ER/PR positive and HER negative, but Ki67 protein is less in case of luminal A and Ki67 is more in luminal B. So there is a subtle difference in luminal A and normal light. Both are same, but only the condition is the luminal A condition is not that much severe, and this normal light is severe in condition. So we have this many different types of cancers, and as I already told, it is having heterogeneous disease and it is differing from individual to individual. So if there is a change in the diversity is present, we have to go for the precision medicine. So already treatments are available like chemotherapy, radiotherapy, and endocrine therapy. And along with that, surgery is also one of the option. And before surgery also, to shrink these cancers or tumors, some therapy will be given that is called as a neoadjuvant therapy. They will shrink the this tumor and then they will perform the surgery. So though there is this many types of therapies are available, and also for the diagnosis also molecular typings are also available, and histopathological diagnosis are also available. This MRI, PET scans are also available, and mammograms are also available, but this is having diversity. So some people, they are not getting good therapy because 46% of the women, they are having this risk of cancers, but they do not require this chemotherapy also. This confusion is only because there is a heterogeneity is present in this, and also there is a confusion in the classification, like stage 1, stage 2. In this stage 3, A, B, C, whether it is in the A or B or C, if there is a some sort of therapy will be present, means there is a different therapy for different stages will be present. So there is a confusion, or there is some clarity is missing. So there is a need for precision therapy. For that, we have to apply the artificial intelligence or deep learning methodologies to correct the staging, to get the accurate staging. Once we know this is the stage, so we can give this type of therapy. So with this intention, we are aimed to do this personalized therapy for breast cancer by developing data-driven algorithms with deep learning technology. So these are the objectives. First, we will collect whatever the radiological images of breast cancer and normal people, and also benign tumors, means they are not cancerous. Some tumors will be present, they are not cancerous. So we will collect all these things. So mammograms, histopathological slides, CT scans, PET scans, and this ultrasound scans of the this breast we will collect, and we will develop as a database. So along with this database, we will collect the case sheets of the patients, and we will maintain as a clinical database. So once we are having images, by using this deep learning algorithms, this is unsupervised ones, because here we are not having any numbers. So by seeing the density or by seeing the cell or cytoplasm or nucleus, the change in this phenotypic characters. So this phenome come into the picture here. Based on the phenome of the breast tissue, we can get some patterns. So this pattern belong to this. So already we are having different types, like molecular typing means luminal A, luminal B, like that. And if the origin of the breast cancer, based on the origin of the breast cancer, it may be lobular or ductal, or based on the nature of the spreading, it is in situ or invasive. So like that, different subtypes are present. So our aim is to predict wherever the patterns are present. Is there separate patterns for each thing is present, or common pattern will be present to some groups? So for that reason, we are planning to identify the patterns for different cancers, breast cancers only. So then if we are having images, and from these images we are having patterns, and from this why we are utilizing here deep learning methodology means for statistical analysis, if there are limited number of parameters, we can apply the statistical methodologies. But in this case, what happens is, so here so many different types of subtypes will be present. So where we can correlate infinite number of parameters can be correlated by using this deep learning algorithm. That is why we selected this artificial intelligence to correlate the images, particularly patterns present in these particular images. Not only with the images, these patterns will be correlated with the clinical symptoms. In this clinical symptoms, I will tell you the details here. And once we are having some patterns, like based on this, all the image databases and clinical databases, we will identify the patterns, and again we will cluster these patterns, and based on this we will do the grouping of the patients. From each group, we will collect the transcriptome or genome, and we will correlate whether there is a correlation is present or not. So this work we are doing. So all of you know about the SWIMS. SWIMS is the tertiary care hospital. with multi-speciality departments. And we are having the histopathology pathology department with 2 labs. One is histopathology lab, another one is clinical pathology lab, and radiology department also we have. Mainly we are having the cancer ward. In these 3 different departments are working in that. One is the medical oncology, radiation oncology, and surgical oncology. So along with this, we are having radiology department. In this radiology, we are having mammograms, CT scans, PET scans, ultra, this UV, everything is available in SWIMS. And every year, we are getting 1,500 patients, new patients, breast cancer patients to the hospital. So every year, we are having, we are getting more than 1,000 mammograms and 350 histopathological slides every year, and we are having the data 25 years, and we will develop a database. So here first we will take, suppose if we are taking a mammogram, and in this mammogram after doing the database, we will see this whether it is a normal or benign or cancerous. So with the help of the radiologist only, we can differentiate, and we will maintain in the form of a database. For all the images, we will do like that. For the histopathology, pathologist opinion we will take, and we will do that. And for the clinical databases, medical oncology person opinion we will take, and we will do. So in the histopathology, again, staging we need to give in the separate database. Histological type means whether it is a lobular or whether it is a ductal, like that we need to give. Lesion type means whether it is a calcification or mass is present, like that we need to give. And again, molecular subtypes means like luminal A, luminal B, or triple negative or normal, like we have to classify, and we have to define in the database. Database means it is a library, and library means if we are not only storing, whenever we want to retrieve, we need to get it back within seconds. That is the database. And again, we are maintaining the clinical database. This information is present in the case sheets, and the electronic health records are also available in SWIMS. So based on these characters, again, one database will be generated. And once we are having this information, then we have to identify the patterns. So 2 different types. One is based on the expert opinion. If the radiology slide means radiology person, with the help of the radiologist, we will define. And another one is without the doctor's opinion, machine learning algorithm we will do. So by comparing these 2, if there is anything wrong with the machine learning, again we will correct, and we need to do the iterations for the time being, and again we have to check the sensitivity. So like that we will learn the system so that it will identify the patterns at par with the expert. Next, once it was identified, correlation will be done with the deep learning algorithms. Once we establish all these things, we can develop a clinical decision support system. Means suppose after 5 years or 10 years, once the research is complete, if we are putting this mammogram in this, so it will read the mammogram histopathological slides, and it will correlate with all the existing ones, because we are giving the data and we are training the system, and it will learn from the data, and it will give some output in the form of a decision. So once we are inserting all these things, we will get some results like this cancer is in this stage, and this drug is suitable, and like that it will give the entire things once this system is ready. And once these patterns are identified, suppose if we identify 10 different patterns, means 10 different groups are present in the breast cancer patients. From each patient, we will take the genome, and we will see the genomic profiles, or we will take the gene expression profiles, and we will identify the targets present in particular group. So suppose if this group will come, so we can directly check that particular gene, whether it is overexpressed or underexpressed. So directly by checking on that, we can give the targeted therapy also for a patient. So this is the first one. This target identification is based on the phenomics, means physiological, we are seeing the morphological characteristics and we are identifying this. And again, this is correlated with the gene expressions. And meanwhile, this is the strategy. So here, this one is first, this one is different databases we are maintaining here. Here we are taking the expert opinion to differentiate whether it is a normal or benign or breast cancer patient. So here we are not taking, in this right side, we are not taking the opinion of the doctor and we are giving the numbers. Here it is machine learning, through the machine learning algorithms, it will read. And finally, the reports will come to here in the objective 3, clinical decision support system. So this images, knowledge-based report means from the doctor, we will get the report. And from the machine, we will get the report, classifier-based report. And again, in the initial stage, there may be some mistakes. So we will iterate and we will correct this and we will do this. If it is at par with the knowledge-based report, then we will integrate and finally we will get the output. And once we are having this, then we will do the gene expression studies. And once this was established, if we are giving the different images like a mammogram or a histopathology slides and this gene expression profiles, so we can say this protein is overexpressed in this particular group. So targeted therapy for the particular patient can be given. So it is the first part of the target identification based on phenomics. And second one is the transcriptomic analysis. This work is also one of my PhD students. She is doing this transcriptomic analysis for the breast cancers. This transcriptomic means the genes expressed in a at a particular time, means in a diseased condition. So for this, normal samples we need, and another one is the diseased samples. So at present, we are working with the microarray data. Microarray means it is having gene chips. Gene chips are present. The cDNA will be impregnated on the slides, and we will collect the mRNA from the patient and also mRNA from the normal candidate, normal person, and we will take 2 slides. And in one slide, this normal person's mRNA will be labeled with CY3 and CY5. 2 tags are present, 2 colors are present. One is red color, one is green color. So this mRNA is mixed with CY3 or CY5. So one is diseased and another one is the cancer patients. So we will take the 2 slides and the normal one we will put on the one slide and diseased one on second slide. So we will get different colors. Means here, if it is overexpressed in the patient, it will be present in the form of red color, and in the normal human being, it is in the green color. So once the hybridization is over, means here it is a cDNA and it is mRNA, so there is a complementarity will be present though. So the hybridization takes place. Once the hybridization is over, so then we will take these 2 slides and we will move on to the one by over the other. So if there is a red color and green color, then it will appear as a yellow color. Means it is present in the normal and also the cancer patients. So if it is only red color, means it is overexpressed in the cancerous conditions. And if it is green, more green is present, means it is more in the normal, means it is down-regulated in this particular cancer patients. So like that we have to do. So that data is available in the GEO database. GEO is maintained by NCBI. This data is present in the CEL forms. So if we are going to this GEO databases, we can get this data. So this is again by using artificial intelligence, we are doing this. So here we will, if we are doing with the artificial intelligence, it is mainly data driven, means we are not taking anything. We are taking only the data and we are analyzing. So we have to give more weightage to the data. If the data is very good quality, then only we will get the good output. So we have to take care for this selection of the data. So here GEO databases, we will get this raw reads. For that, first we need to check the quality. This quality means if these reads are correct or not. If they are curated, it's okay. If they are not curated, we have to check. Though it is a curated, we have to check the data, whether it is curated or not. So this for this fast QC, this program, if we are running in the R, so we will get some information, like we will get Phred scores. So if it is, these reads are having 100 to 150 in length. So in this, if we are getting the Phred score of 30, means 99.9% of the bases are correct. If the Phred score is having 20, means 99% correct. And if it is 10, it is only 90%, means for every 100 residues, 10 residues are wrong, means we cannot take. So generally we will take 30 to, in between 30 to 20 score will be considered as a good reads, and we will take the data for the further analysis. Again, once we have a quality data with our hand, in our hand, then we need to align with the human genome. So there are so many genomes are available. So recently published one in NCBI is GRCH38. This genome is why we need to align means. So if you take this in human beings, different tissues will be present. Some isozymes will be present, like LDH. This is lactate dehydrogenase. If this lactate dehydrogenase behaves differently in liver and differently in brain. So to consider all these things also, we have to align with this GRCH38. Means this will use the splice site aligners. Means the gene will be same. Based on the exons, there is some changes will be present in the splice sites, and there will be subtle difference will be present. So to consider whether there is a change in the brain or whether change in the lung, we have to consider all the isozymes. So we need to align this with GRCH38 reference genome. Once the alignment is correct, then we will go for the normalization. Normalization means these values, these transcript values will be from minus 1000 to plus 1000. So if these values are not matching, it will cause some difference. So by taking this, and we have to normalize the data. So in the statistics, this is the routine process. Normalization we will do. So once the normalization is done, then we will run the programs to see the differentially expressed genes. Differentially expressed means there are two different types, whether they are overexpressed or underexpressed. So these genes we need to identify. Then once the differentially expressed genes are available, and again we need to check for the gene ontology enrichment, pathway enrichment, protein-protein interaction networks, and functional module analysis. And these things we will see in the coming slides. So this is our work. In the GEO databases, the databases related to breast cancer are 9,356. But after curating all these things, we have selected only 155 Among this 155, again 81 data sets belong to cancer clinical samples. So we have selected these clinical samples. In again each data set, there are different number of normal patients and different number of diseased patients will be present. So in this study, we have selected 2,288 samples, among which 307 are normal and 1,981 are diseased samples. So these things we have taken, and we did the quality control, and also we aligned with the human genome, and again we normalized. After the normalization, we did this differentially expressed genes. Once the differentially expressed genes are there, then we have to check for the p-value. P-value if it is less than 0.05, and fold change value if it is more than 2, then we will consider. Sorry, this sign is wrong. If the fold change value should be more than 2, then only we will consider this as a differentially expressed genes. So among all these things, as I already mentioned, this is a heterogeneous disease. We have considered all these patients to find out the common drug targets, but we are unable to find a common drug target. So what we are doing is, based on the molecular subtypes like luminal A, luminal B, normal like triple negative, and basal like cancers, we will separate the these samples into based on the molecular subtypes, and in each subtype we are planning to identify the genes, differentially expressed genes. So these are the reads. So this is the block heat map generated in the DEG databases in the transcriptomic analysis. So here it gives the red color indicates the overexpressed ones. So then we need to plot we will get so many in the 30,000 genes will be present. In this 30,000 genes, we have to select the differentially expressed genes. So in this volcano plot, if you see this log FC value, if it is more than 2, then only we will consider, or less than -2, then only we will consider. So this differentially expressed genes, by plotting in the volcano plot, we have to select. And this red color and blue color means overexpressed ones and downregulated ones. So once we have these DEG databases, again we have to submit to the gene ontology analysis. So in this gene ontology, we will get 3 different types of information. What is the biological process, what is the molecular function, and what is the cellular component. So like that, we will get the more details about the differentially expressed genes. So once we get this, again we need to identify the pathway of this particular differentially expressed genes. So it will, by submitting to the KEGG or David database, we will get the pathways involved in this. And main important thing is this interactome. So if we see the normal patients and also the diseased patient, cancer patients, these interactions are different. So the recent study is telling that if there is a different interactions, the protein is interacting with other protein, it causes some cancers. Like that, it is important to identify the different patterns. As we are doing this with the molecular subtypes, these plots we will take and we will correlate with all these things by using these deep learning techniques. So here also the red color means overexpressed and the blue one means it is less expressed ones. Again, we need to run this CytoHub, means it is Cytoscape is a freeware, and in this software, once we are having this upregulation and downregulation, so this CytoHub, this tool, it is having 12 parameters. In this 12 parameters, if this differentially expressed genes are having more than 6 score, means minimum 6 parameters are satisfied, then only we will consider this as a drug targets. So with this, we are having the drug targets from the phenomic data and also the transcriptomic data. We are having the drug targets. So first step is over, target identification. Once we are having a target, then we have to check for the structure. If the structure is available, then we can download the structure from the PDB databases and we can analyze. If the structure is not available, we have to go for the homology modeling. This homology modeling means if the structure is not available, we have to check the homologue from the PDB databases. If it is having more than 30% identity, then that structure will be downloaded, and by aligning with this existing one, we can generate a model. Once the model is generated, again we need to validate. Means whether the stereochemistry of the amino acids are obeying the rules or not. So based on that, dope score will be given. That energy is less means it is having more stable. Generally, we will generate 100 models for the homology for the drug targets. Among the 100 models, whichever is having the lowest energy, that model will be taken. And again, we will do the ProCheck analysis. In that stereochemical analysis, stereochemistry of the each and every residue will be checked. If it is stereochemically stable, then we will see the next step of validation with the superposing command with the already existing PDB structure, and this one we will superpose, and if there is a less RMS deviation, and then we will consider that model. And if the structure is having the ligand also, we will do the modeling along with the ligand. And once the model is there, then we need to do the protein preparation. If the model is, the structure is available in the PDB database, directly we can download and we can do the protein preparation, this wizard in Schrodinger software. And once we are having the 3D structure, and once we know the active site, we can generate a grid on the active site. Why we are taking the active site means, if we are taking a protein molecule, it is difficult to block that protein activity. If we are taking an enzyme, this enzyme will be having active site or allosteric site. So we have the known information about the target. By targeting this active site means, if we are targeting the active site means, if it should be, the molecule should be having more affinity than the substrate, then only competitive inhibition will be present. Means in the presence of substrate also, this our compound will goes and binds with the active site and it blocks the activity. Or allosteric site means, if there is a ATP molecules and GTP molecules are present, these are also important for the functionality of the protein. If we are blocking this also, we can stop the functionality of the protein if this protein is in the overexpressed condition. If the protein is in the down regulation, we need to activate. So these are the two different things we need to do. So once we are having the protein structure and the area where we need to block the function also, if we know, then we can do the docking studies. And also for the ligand discovery, again there are of three different things are present. Means if we are having IC50 values, biological activity of the protein are present, then we take that IC50 values and we can convert that into PIC50 values, and we can generate a QSAR model. Based on the QSAR model, we can screen in the available databases. Or if we are having the co-crystal structures, if we take the EGFR molecule, we will be having more and more co-crystal structures. Based on that, we can generate the E-pharmacophore models and we can screen in the databases. And if the crystal structure only available without the ligand, then we can go for the virtual screening. So these are the three screening mechanisms. So we can screen based on the QSAR model or pharmacophore model or virtual screening. So once we are having a target molecule with a definite grid, then we have to search in the literature for the molecules or drug bank databases like that, or PubChem. We can check the molecules for this particular target, and we have to search for the similar molecules present in our in-house library. We are having 28.5 million small molecules in our lab. So first we will screen these compounds in the in-house library. Means this process is called as a virtual screening, so that we will get some ligand molecules. This is called as a ligand data set. This ligand data set with the grid generated protein molecule, we will perform the docking studies. Generally, we are doing with the Glide because we are having the Schrodinger software. And this Glide docking is of 3 different types. Generally, it is a rigid receptor docking. Rigid receptor docking means it is the molecule is rigid and the ligand is also a rigid. So it is called the molecule is flexible. So receptor only rigid. So it is called as a rigid receptor docking. In this again there are 3 stages. First one is the HTVS, SP and XP. HTVS means high throughput virtual screening. SP means standard precision. XP means extra precision docking. So the names indicates high throughput means here we are having 28.5 million. So we have to screen them very fast. So we will give the less negative scores and we will delete it from the list. SP means here we will give the some more weightage, and in XP we will give more weightage if there is any steric hindrance. Like that also we will give negative values and we will give the scores. Once we are having this XP, then we will check for the MMGBSA, means free energy calculations we will do. So this is the ordinary docking, rigid receptor docking. So here we are not considering the solvent model. So here MMGBSA means here we will calculate the where the protein is present, whether it is present in the cytoplasm or whether it is present in the lipid layer. Like that we can calculate, and surface area also we will consider for the docking studies. MMGBSA means molecular mechanics, generalized bond, surface activity relationship. We will do this docking and we will get the free energy. The final score is in the form of free energy. So once we will get this one, again we will go for the quantum polarized ligand docking. So in this, generally we are calculating the scores based on the molecular mechanics. To calculate in quantum mechanics, it requires a lot of computational speed. So what we are doing is only for the active site region only, we are applying the quantum polarization and we are checking in this condition whether this ligand molecule is docking with the protein molecule or not. And once we are getting the QPLD scores with prime MMGBSA, means free energy calculation of QPLD docking, then we will compare the lead molecules with the not lead, that are ligand molecules with the existing inhibitors. Once they are better than the existing ones, then it is called as a lead molecule. So these lead molecules again we will subject to the induced fit docking. Induced fit means previously it is a flexible, not a flexible docking, it is a rigid receptor docking. So here we are putting this receptor as a rigid molecule and this ligand molecule as a flexible. So here what we are doing is, instead of giving the flexibility to the entire receptor, we are giving the flexibility to the side chains of the active site region, and we are doing in this condition our lead molecule is binding with the protein or not. So then if we are getting the good score in the induced fit docking also, then we will propose as a lead molecule. So this docking is only one conformation of the protein and ligand complex. For that, to check in different conformations, we need to do the molecular dynamic simulations. Generally, in our lab, we are doing 300 nanoseconds molecular dynamic simulations. So with this, we will get 30,000 different conformations of protein and ligand. In each and every conformation, we can see the how energy of the complex is present and what are the RMS deviations, RMSF calculations. We can see all these things, and interaction plots also we can see. And finally, we can propose the lead molecules. So once we are having these lead molecules, this lead molecule is the molecule which is present in the database. So to get the patent, we cannot go for that particular molecule. So we need to develop a novel molecule. For that, we have to go for the core hopping methodology. So before this also, I forgot to tell one thing. So once we are having, after the QPLD docking, if we are getting the good molecules, then we have to validate these models. Validate these lead molecules means here we are taking the 100 decoys and these lead molecules and the existing inhibitors we will combine and we will form into a database, and we will do the docking studies. So in this, if we are getting a good decoys separately and active molecules separately, and where the lead molecule is positioned also considered, and we will propose the lead molecules. So this is the overall story. So these novel molecules will be generated by using core hopping method. So this is the molecular dynamic simulations. So here we will get different 30,000 different conformations, and each and every conformation we can see the RMSD, RMSF values. And here we can also see this is the GBR, means it is the target and the PDB ligand, and this one is our proposed lead molecule. If you see this, we are having more interactions with our lead molecule. So we can propose this. Not only with the scores, we can also see the interaction plot. And this work is published in this Journal of Molecular Graphics and Modeling. And another one is this Biomedical Engineering Society, this cellular and molecular bioengineering also we published one paper. And if the structures are if the ligand molecules are having IC50 values, suppose if we are having 100 different compounds, and we can take these 100 compounds into 65 trainee sets and 35 test sets. So based on this, IC50 values will be converted, and again we can do the regression analysis, and finally we can generate a QSR model. So this R means this is the ring models, and here this QSR or pharmacophore model, we will be having R means ring, and D means hydrogen bond donuts, H means A means hydrogen bond acceptors, N means negatively charged. Like that we will be having these things. So if we are having the QSAR model, we can screen this model in the virtual screening. So this is called as the QSAR based screening. This QSAR based screening is possible only if we are having the biological activity of the compounds. So once we are we did it for EGFR, and this is the PDB structure, and this PDB ligand, this gefitinib is having minus 9.95 kilocalories, but our proposed model is having minus 12.682. In case of this free energy calculation also, we find 98.4% 4 kilocalories, but our molecule is having minus 130 kilocalories per mole. So we proposed this, and this work is also published. And if we are having more crystal structures, and these co-crystal structures, more structures with co-crystallization, and these co-crystal structures are also diverse in nature. If the same structure is present, same ligand is present, we will not consider that, among which we have to select whichever is having less R values. So less resolute structure we will take. So like that, for JNK1, we are having different E-pharma diverse ligands, 7 diverse ligands. We have these 7 diverse ligands. From this, we generated the common pharmacophore model, means among which these 7 are representing here. So this CPH model means if we are taking these 2 models, we have to retrieve these 7 ligands, then it is called as the CPH model, common pharmacophore model. For screening, instead of 7 pharmacophore models, we can take the pharmacophore models and we can screen, and these similar ligands we will get, and again we have to filter by using the LeakPrep and ADPIC. And with this HTVS screening, we got this many, and with the SP65, and finally by comparing with the existing inhibitor, we find only 5 hits. So this is the ROC plot, validation of the lead molecule. So here we are taking 1000 decoys and 5 these hits, and these 7 ligands already existing in the databases. We will run this ROC plot, and all are having these good values, and decoys are separately separated, and these actives are separated. So our lead molecule is validated as a good molecule. And this work also published in Journal of Receptor and Signal Transduction. And this is the core hopping method. Core hopping method means for this core hopping, we need to develop a novel molecule. So for these parent compounds, we have to take the existing ones and also the lead molecules, and we have to add the side chains and some different ring moieties. So these fragments, by adding these fragments, and again, if it is having, it is following the Lipinski's rule and ADME properties and synthetic capacity of the compound, then we will propose this as a lead molecule, novel molecule. And again, we will check in the gene database, or if the chemical databases also we need to check. If it is not available in that, means our compound is a novel molecule. So this work is published in Computational Biology and Chemistry. So with this artificial intelligence, so previously it is a linear fashion. It is a circular fashion. So if we are having one target, and these interactions we can study, and we can collect the molecules, and among this molecule, whichever are not following Lipinski, we can eliminate, and the compounds which are having the possible drug-like compounds, we will put in a database, and these compounds can be used for any type of diseases. Like right now we are using this repurposing drugs for COVID-19. So this remdesivir is previously developed for this malaria. So now if we are having this type of database with the druggable molecules, and we can, if we are having a novel target, and by going through this cycle, we can propose a molecule as early as possible. This speed, generally we will take 15 to 20 years to get a molecule. For this COVID-19, we got molecules within the three months because of this methodology. So with this, I came to the conclusion, and there are some challenges in this artificial intelligence. First one is this is data driven. So our data should be very quality data should be taken, because all these are the data driven. 80% of the results is based on the input data, and only 20% weightage will be given to the algorithms. And in case of breast cancers, as we have seen, different types of breast cancers are available. So by doing the statistics, this many parameters we cannot correlate. So by using the deep learning algorithms, this is possible to see the infinite number of parameters can be correlated by using deep learning algorithms. And generating new hypotheses is also possible, because if we are doing the de novo designing with the core hopping, and we can get the novel molecules also. And it is reducing the time also, because we are having a good computational speed. Previously, we used to have CPUs, central processing units, then graphical processing units. Now we are using TPUs. So it is having more speed in the computation. So we can reduce the time also for getting the results. And there should be a mind change in the research culture also. Previously, if there is a bench to bed, should be replaced with a computer table to pharmacy racks. So the final goal is we have to combine the big data. Big data is available with our omics, that is genome, proteome, everything is in the enormous data is present. And to analyze this, we need to have an artificial intelligence. With the human brain, we cannot analyze. So we need artificial intelligence. By amalgamating the big data with artificial intelligence, we can get the good decisions. And finally, our goal is to get quicker, cheaper, and more effective drugs. So this is the take-home message. If we are mixing the mind and machine, if we are doing this cyclic process, first computational scientists, they need to design, and the synthetic chemist, he need to synthesize, and biologist, he need to test, and again by using the computational person, he need to analyze. This cyclic process should go, and we can get the good decisions. So the integration of mind and machine is required. It is the need of hour. With this, I conclude my talk. Thank you very much for your patient listening. I'm happy to take questions.
Yeah, thank you so much, madam. And now the session is open for questions. So we got one.
Ma'am, there is a question from Mr. Keshav Moharir.
Yeah.
What is the difference between big data and metadata?
Big data means we are having enormous data in diversity. Metadata means, suppose if we take pathogens, suppose if we are working with the pathogen, if we are taking one genome, it is one genome, it is a big data. Metagenome means if we are taking the entire gut genomes, means in gut so many microbiome will be present. Means metadata, meta means it is having n number of organisms. Here big data means it is also a big data, but if we want to specifically identify the drug targets, we will take single ones. Metadata is the best example is our gut microbe. Here also we will get the big data.
So if there are any questions, please unmute yourself, and you can also ask or leave them in the chat box so you can ask, ma'am.
Pooja, somebody raised the hand. You just see.
Suchitra, it seems.
Suchitra, do you have any
They can
doubt?
unmute themselves and ask, ma'am.
We are getting so many appreciations for the session, madam.
Thank you.
Suchitra. Okay.
If there are no questions, we can proceed to vote of thanks, madam.
Okay, Ra.
Okay, so it's a matter of pride for me that I'm proposing a vote of thanks for this session. Dr. Amineni Uma Maheshwari madam for sharing her research work and knowledge with us. Madam has focused on the classification of machine learning methods and their applicability to drug development process. We got to know the new term, that is omics, stands for, what it stands for, and what are the different techniques of omics. Presentation was very loose.
Shaheen?
Disease stages, as well as for the personalized and ideal therapy for the cancer. We got to know that what are the different types of cancers, madam. Previously, we used to know only one type of breast cancer. We don't know how many these types of cancers are present.
Shaheen, your voice is breaking and it is not audible.
I can say that it is a perfect blend of theory and practice of AI. In this session, you have clearly demonstrated the theoretical approaches and how they can be utilized. Have got lot of information and knowledge from your session, madam. I would like to thank all the participants, and I would like to thank our organization for giving me this opportunity to be with you, madam. Thank you so much.
Thank you, Dr. Uma, for accepting our invitation. And I thought that this is the right platform to acknowledge you for your help during our talking studies of our students of our institute. And my heartfelt thanks to your presentation. It is very, very informative and thought-provoking. And you are the one of the person who stood first to help all of us, especially for our institute. And we hope same in future also. Thank you. Thank you very much, Uma. Thank you.
Thank you, Sridevi, for making a part in your FDP program. Thank you very much.
Thank you. We'll see you later. Huh? See you.
Okay,
Bye.
okay. Bye. Bye. Thank you.
Thank you. Dear participants, afternoon session will start sharp 3 o'clock. Okay, join at 3 o'clock. Thank you.
Hello, madam. We got hello.
Shaheen.
Madam, evaru maatladutunnaru madam. Back.
Participants, tell me. Hello.
Sir, your voice is not clear, sir. Your voice is not clear.
Hello.
Bhasme, sir, we are unable to listen your voice.
Hello.
Madam, majority of them, they will ask for PPTs. You can ask Uma madam to share her slides, madam.
I will ask. Okay. I'll request the madam. Okay.
Evaru maatladutunnaru madam venuka kani clear ga ravatledu voice. They are trying.
Okay. That is the summary, no? They need PPT. Anything else?
Ante madam. Manakaithe vinipinచట్లేదు voice.
Okay.
Arifa Begum is asking, please plan for workshop also.
On? Actually, this
AI.
is for we planned for workshop only, but due to the COVID, it is converted into
Online
such type
platform.
of training program. Okay. In future, we will do that. And with all your cooperation.
Oh.
मिक्सर मिळाले मला जेवायला सोबत. I hate to eat alone.